With one of the most important topics of human anatomy and physiology first, that is the sliding filament theory. Means in this theory we will be going to discuss how the muscles contract, how the skeletal muscle contraction occurs in our body. So before going to the sliding filament theory, I will be explaining the organization of the skeletal muscle, how the skeletal muscle is organized in our body. So I will start. I have made the diagram here. So I will start with the first one. For example, here this is a bone. So this bone is attached with the muscle. So this here is a muscle. So the bone is attached with the muscle, and the attachment is through tendon. So tendons attach the bones to the muscle. So this is a bone, and this bone is attached to the muscle through the tendon. Okay, so I will number it here. So this is the muscle. Okay, so the membrane which is present outside the muscle, this outside membrane of the muscle, this is called as epimysium. Outside membrane of the muscle, this is called as the epimysium. Okay, so muscle. So if we will go inside this muscle, so inside this muscle there is a one more bundle which is called as fascicle. Small bundles which are present inside the muscle, those bundles are called as the fascicle, and this fascicle, it is a tissue. So here muscle, we can say it is an organ, and this fascicle which is present inside the muscle, this is called as the tissue, and the membrane which is present outside this fascicle, this is called as perimysium. This membrane is called as perimysium. So bone is attached with the muscle by tendons, and inside the muscle there is presence of a bundles which are called as fascicles. Inside fascicles there are much more bundles which are called as muscle fibers. So I will go to this one. There is presence of muscle fibers. So this is a muscle fiber, or we can say that these are the cells. So this is a cell. Okay, and the membrane which is present here, this membrane is called as endomysium. So three membranes: epimysium, perimysium, and endomysium. So inside, first there is a muscle. Inside muscle there is presence of fascicle. Inside fascicle there are small tubules which are called as muscle fibers or cell. So inside this cell there is presence of sarcoplasmic reticulum. So I will write it here, SR. So this is the presence of the sarcoplasmic reticulum, and there are transverse tubules also present. Transverse tubules, these are important when we will study about the contraction. Okay, so inside this muscle fiber there is presence of myofibril. So this is the fourth one, which is myofibril, and inside those myofibrils, myofibril, it is an organelle. Inside those myofibrils there is presence of the proteins. So these are the proteins. So in short, you can remember. So this is the fifth one. So this is a muscle. Inside muscle there is presence of fascicles. Inside fascicles there is presence of the muscle fibers. Inside muscle fibers there is presence of myofibrils, and these myofibrils contain the proteins. And which two proteins are present mainly here? Actin and myosin. So the proteins which are present are, one is actin and second is myosin. So these are very important for the process of the contraction. Okay, so important for the process of the muscle contraction. So this is the organization of the skeletal muscle, how the skeletal muscle is organized in our body. Now if we will go to the myofibril part, because that is important for the contraction process, so I will take this myofibril part out. So I am going to draw here the diagram of the myofibril part. So if we will go to the myofibril contains the actin and the myosin filaments. So it's like this. So these spots which I am drawing here, these are the actin. So this is the inside part of the myofibril. So if I am going deep inside the myofibril, so myofibril, it looks like this. So this is the organization of the myofibril. So this is the myofibril. Okay, so these parts, these are called as the actin, and inside actin there will be presence of myosin. So these are the actin and the myosin filaments. So this one which is present here, this is called as the actin. Actin is thin, whereas this myosin, this is myosin, myosin is thick. So there are thin and thick filaments which are present, okay, and this all is present inside the myofibril. Okay, so this is inside the myofibril. I have drawn it here also. So this is drawn inside the myofibril. So these units, these are called as the sarcomeres. So these, I will draw it here. So if I will go from this part to this part, this whole will be called as sarcomere, which are called as the contractile units. So this is the contractile unit of the muscle, which is called as the sarcomere. Okay, and this line here, this is called as the Z line. This is second Z line. So between two Z lines there is one sarcomere. Okay, now the area where only the actin filaments are present, okay, the area where only the actin filaments are present, that is called as the I band. For example, this part here, only the actin filaments are present here, this band will be called as the I band, and the actin and myosin where they overlap, that is called as the A band. This one is A band. So the band where the actin plus myosin overlap. So overlapping of actin and myosin, that is called A band. I band. In I band there will be presence of only actin. There is one more band which is called as H band, where there is presence of myosin only. Myosin only. So this is the structure of the myofibril. If you will go inside the myofibril, so I told you there is presence of two proteins, one is actin, one is myosin. So I have drawn the structures here. So this is these are the two sarcomeres which I have drawn here. Every sarcomere, it contains, see, the thin filaments which are called as the actin, while as the thick filaments, these are thick in nature. Okay, so these thick filaments, these are called as the myosin filaments. There are two, these lines, these are called as the Z lines or zigzag lines. These zigzag lines, see, you can see here, this is a zigzag line, this is called as the Z line. So the area where only the myosin is present, this is called as the H band. Between two Z lines, it will be called as sarcomere. The area where the actin and the myosin overlap, that is called as the A band, and this band where only actin is present, this is called as the I band. So this is about the organization of the skeletal muscle, how the skeletal muscles are organized. Now we can go to the second part, which is the sliding filament theory, or how the contraction occurs. It will be easy for us to understand that process. So I will go to the next part. So sliding filament theory, for that sliding filament theory, I will be drawing one diagram first. So this one, and with this, so this is a neuromuscular junction here. So this, I have drawn the neuromuscular junction. This is the nerve ending. This is nerve ending. Okay. And how it will start? The first thing will be the action potential, which will come from here. Action potential, or what we call it as nerve impulse. This action potential will reach here. So here there is a presence of acetylcholine, which is present in the synaptic vesicles. So these are called as the synaptic vesicles. Synaptic vesicles, which store the neurotransmitters. So here the neurotransmitter which I am explaining you, that is an acetylcholine, ACH. Acetylcholine is a neurotransmitter which is formed by the combination of choline and acetyl coenzyme A. Okay, so action potential, it will reach here. This action potential, what it will do, these are the presynaptic vesicles, or we can write presynaptic vesicles. Okay, so what will happen, there are other channels also. For example, here there is a presence of the calcium channel, Ca2 positive. When action potential reaches here, this calcium, it moves from outside to the inside. When it moves into the inside, this synaptic vesicle fuses with the membrane. When it fuses with the membrane, there will be a release of acetylcholine. So this is the acetylcholine, and this is the neuromuscular junction. Neuromuscular junction. So here there is a presence of now these neurotransmitter will be released, and which type of neurotransmitter here is released? Acetylcholine. Okay, so calcium enters inside the cell. First the cell was inside, from the inside it was negatively charged. Now when the calcium enters, it becomes positively charged. Okay, so calcium enters from here and here also, Ca2 positive, and outside there is high concentration of sodium. So outside there will be high concentration of Na positive. But during action potential, calcium will enter inside this. Okay, so there is entry of calcium inside inside the nerve ending, and that calcium triggers the release of the acetylcholine from the synaptic vesicles. So now here is the presence of the membrane, so I will draw it completely. This is the transverse tubule. Minutes before we studied about this transverse tubule. So here there is the presence of the sarcoplasmic reticulum. So this is sarcoplasmic reticulum. I will write it short at SR. When this acetylcholine reaches here, okay, so this acetylcholine reaches here. So here there is a presence of ligand gated sodium channel. This is ligand gated sodium channel. So it will be closed first. Okay, this ligand gated sodium channel, it is closed first. When acetylcholine reaches here, the acetylcholine binds with this ligand gated sodium channel and causes the opening of this gate. So this gate here will be opened. Okay, there is opening of the gate, and this sodium ions, sodium ions will move inside, and inside now the charge will become positive sodium ions. Okay, and this action potential which was coming from the neuron, this action potential will propagate through here, and it will reach the transverse tubules. So this is action potential. Okay, so this action potential which will reach the transverse tubules and to the sarcoplasmic reticulum. And sarcoplasmic reticulum, what is the function of the sarcoplasmic reticulum? It stores calcium, Ca double positive. So it will store the calcium. Okay, so when this action potential reaches the sarcoplasmic reticulum, it triggers the release of Ca2 positive, because calcium is responsible for the contraction. So from here action potential, it causes the sarcoplasmic reticulum to lose the calcium or to release the calcium. Okay, so calcium is released here. One more thing which we have to remember here is that when this acetylcholine does its function, after that what happens to acetylcholine? There is one enzyme here which is called as acetylcholine sterase. This is the enzyme here, acetylcholine sterase. It breaks down the acetylcholine into acetate plus choline, and this acetate plus choline then goes back into the presynaptic vesicles. It goes back into the nerve ending. Okay, so it goes back there. So acetylcholine also, here there is presence of the receptors. There can be nicotinic or muscarinic receptors, either N receptors or M receptors, which we will be studying in the pharmacology. So nicotinic receptor, they can be NN or NM, and muscarinic receptor M1, M2, M3, M4 and M5 receptors. Okay, so there is presence of various receptors here. So I will again explain you this thing. So action potential, when action potential reaches here, this action potential causes the calcium from outside to come inside the cell, and here inside the cell there is presence of the acetylcholine which are present inside the synaptic vesicles. Calcium binds with these synaptic vesicles and fuses with the membrane, which causes release of the acetylcholine in this The synapse are in this gap between this, which is called as the synapse. Okay, and this acetylcholine then reacts with or then binds with the receptors, either nicotinic or muscarinic receptors. Okay, and then it causes the action potential to reach the sarcoplasmic reticulum, where there is release of the calcium from the sarcoplasmic reticulum. So calcium is released from the sarcoplasmic reticulum. Now what this calcium does? Okay, now again we will go to the second diagram. For example, first thing I will write here, off muscle. Okay, so first thing what happens, that already we have explained about the actin and the myosin. For example, so here I have drawn the structure. So this structure consists of, so here there is presence of the actin first. So these are the actin filaments, and this is the myosin filament. So this actin, what it has the presence of the troponin and tropomyosin. Troponin is present there, and one more thing which is present here is tropomyosin. Okay, so this first step I called it as off. Off means muscle here is relaxed. So this is a relaxed muscle which you can see on the diagram. So that this muscle which you can see here, it is a relaxed muscle. So there is presence of the myosin. These are the myosin binding sites, which are called as the binding sites. Here this is presence of actin, this is troponin, this is tropomyosin. So what happens in the next step? We'll go to the next step. What happens that, so here the calcium which comes from the sarcoplasmic reticulum, this calcium, it will bind with this calcium. Okay, this calcium binds with troponin, which is present here. So this calcium binds with troponin and pulls the tropomyosin. See why this muscle is up here? Because there is presence of tropomyosin. You can see this line. You can see this line here. This is the tropomyosin. So when tropomyosin is present, the muscle will be in the relaxed state. When the tropomyosin will be released, then the troponin, the active sites of the troponin will be exposed, and then the contraction will start. Okay, so calcium from the sarcoplasmic reticulum, it binds with the troponin, which pulls tropomyosin from the binding sites. So it pulls tropomyosin from binding sites. So this tropomyosin, it's pulled back from the binding sites. So what happens in the second step is that, so this is the step number second. So this is the actin. Okay, now tropomyosin, it has been pulled from the active sites. For example, it has been removed from the active sites, and now the troponin is exposed. Okay, in the second step, this troponin here is exposed. For example, this is the presence of the troponin. Now this troponin is free. This troponin is free. To this troponin, what will bind? Calcium, Ca2 positive, which came from the sarcoplasmic reticulum. So now this muscle is called as the on muscle. I will write here. So this is on muscle. First step, the muscle was off. Now second step, the muscle is on. Why the muscle is on? Because the calcium from the sarcoplasmic reticulum, it removed tropomyosin from the binding sites. So troponin is exposed. Calcium will now act on the troponin. Okay, now the third step, what will happen? This third step we call it as grab. So there will be grabbing. Means myosin heads form a cross bridge. Myosin heads form cross bridges. How they form the cross bridge? You can see here. For example, again, this is the actin, and now we have the myosin. See, they form the cross bridge with the actin. So this is called as the grab. So what here happens, that the myosin heads, they grab the actin heads. So this step is called as the grab. Now the fourth step, what happens in the fourth step? We will go to the fourth step. So it is grab. Now fourth step, we call it as power stroke. In power stroke, what will happen? Just like a sweeping. See, you can see the direction of myosin filaments now. These are the actin filaments. So this is actin and these are the myosin heads. Now they are grabbing this actin and they are pulling it. Okay, that's called as the power stroke. Okay, now after the power stroke, when the myosin heads, when they bind with the actin and pull it, so this step is called as the power stroke. Now what will happen in the next step, in the fifth step? Right here, the fifth step, release plus reset. Release plus reset. Now the myosin heads, they will be released and again they will reset. Okay, so there will be release and reset, again release and reset, and then after release and reset, what will happen? It will again go to the first step, release and reset. Again it will go to the step number second. Then this process will continue, the contraction process will continue. So when the contraction will end at this last step, what will happen? Turn off. Sixth step will be turn off. So calcium is pumped back to the sarcoplasmic reticulum, Ca2 positive, which was present on the troponin, so it will go back to the sarcoplasmic reticulum. Okay, so calcium goes back into the sarcoplasmic reticulum. So as long as calcium is present, the steps will be repeated, 2 4 3 5 2 3 4 5 2 3 4 5, those steps will be repeated. Okay, so again I will explain you. Here there was presence of tropomyosin, there was presence of the troponin. Okay, you can see here there is presence of troponin, there is presence of tropomyosin. So tropomyosin, what it does, it blocks the active sites of the troponin so that calcium cannot act on the troponin in the relaxed form. Okay, so when calcium is released from the sarcoplasmic reticulum by the above process, what happens that this tropomyosin is released, troponin, the active sites of the troponin, they are exposed to the calcium. So calcium will act on this troponin. When calcium acts on the troponin, that means muscle, it gets activated. So we can say it as on muscle. And third step is grab, means myosin heads, they will grab the actin heads and then they will pull it, and this which is called as the power stroke. Then there will be release, then release of the myosin heads, then again it will reset, then it will repeat all those steps until the contraction process occurs. So when the contraction stops, the calcium goes back into the sarcoplasmic reticulum, then there is stoppage of the contraction process. So you can see here in the muscle contraction, what will happen? This will be contracted. Okay, so this sarcomere, the length of the sarcomere, it will be shortened. So I can draw it here. After the contraction, what will happen? So it will become short. You can see the length here. So because of the calcium, it has shortened and muscle has contracted. So this is the process of the muscle contraction which occurs in our body.